So what kind of solution will your cells be happiest in? Isotonic. Why? The same concentration of dissolved substances in both solutions inside and outside cells. Therefore, water, and water is going to play the big role in tonicity. Water is doing what? Coming and going at an equal rate. Yes? If I put a solution into your body that is hypertonic, that solution has a higher concentration of dissolved substances in it. What's going to happen to the water inside your cells? It's going to move out faster than it moves in. Cause your cells to shrink up. What's that fancy word? Begins with a C. Crenate. That's not good. And then the opposite of that is a hypotonic solution, a solution that has a much lower concentration of dissolved substances than we find inside the cell. Water's going to go try to fix that by doing what? Rushing into the cell, and eventually what's going to happen to the cell? Boom! It's going to do what? Don't say explode. This is a science class. Lyse. It's going to lyse. Yes? So we sort of finished talking about passive processes of movement across the plasma membrane. Give me examples of passive movement across the plasma membrane. Simple diffusion, somebody said. Very good. What else? Osmosis. Carrier-mediated, facilitated diffusion. Channel-mediated, facilitated diffusion. Yes? All of these processes are what? Passive. Passive meaning what? No energy required for movement. Energy in the form of what? ATP. Remember our energy molecule, ATP? So understand passive movement, understand tonicity and water's role in that movement. The other types of movement we can have across a membrane require what? Require energy to happen. Guess what we call these processes? Active transport. And active transport is using ATP, using energy to do work, to move things across the membrane. Many of these things are being moved against their will. For example, the diagram that we see up here is talking about something called the sodium-potassium pump. We only see the sodium, I think, in this. Oh, no, there's potassium there, too. Now, remember what molecules want to do naturally? Where do they want to go? Yep, they want to be away from each other. They want to go from a high concentration to a low concentration. They want to follow their what? Concentration gradient. But these guys are going to move them in the opposite direction, against their will. Sodium naturally wants to go from a high concentration to a low concentration. These guys are going to push them from a low concentration to a what? High concentration. Does that take energy? Yes, it does. Why the heck does a cell do that? When we talk about action potentials and depolarization of cells, electricity, basically, when we discuss the nervous system, we'll see that these pumps are going to play a major role in nerve or neuron transmission. When these pumps stop and let everybody do what they're supposed to do, go with their concentration gradient, the charge inside the cells, which is normally at rest, more positive on the outside, more negative on the inside is going to change. Once that happens, we're going to set off a series of events that's going to transmit electricity down the cell to the next cell. So these proteins, these protein pumps that perform active transport are very important for normal cell function. So we have primary active transport, and an example of this is the sodium-potassium pump. Again, this is an integral protein that we find sandwiched in the plasma membrane. Sometimes we refer to these guys as ATP aces. Why do you think we call them that? Well, what's ATP? Energy. ASE on the end of a word. Enzyme. What do enzymes do for a living? Help with, they can break things down or they can build things up. They're going to help with chemical reactions, right? They're catalysts. Now do you see why sometimes we call them that? Anybody? Anybody got a head nod? Nothing? I got nothing. Secondary active transport is, again, one of these guys, proteins in the surface of plasma membranes that help push things against concentration gradients. So we have a whole bunch of different pumps. We have the sodium-potassium pump. We have the sodium-glucose pump. We have all kinds of protein pumps. Again, performing different functions for the cell, allowing things to come in and out of the cell depending on the cell's need. So vesicular transport. If I have a whole bunch of stuff I got to get in and out, instead of putting it through a tiny little protein pump, I have the ability, kind of like an amoeba, to take parts of my plasma membrane and wrap them around things and create a little package. Think of this as trying to get on a submarine when it's underwater. Okay? How do you get in a submarine when it's underwater? You just go down, knock on the hatch, somebody opens it up, right? You just go in. They'd be really angry with you if you did that. Why? Because all of the water would rush into the sub and kill everybody, right? So how do you get into a submarine when it's underwater? Do you know? You can. Yeah, they have a little chamber. So you open that little chamber's hatch. You have to tell them inside, make sure you shut your side. You get in along with all the water. You shut your hatch, and then a pump does what? Pumps all the water out. Then you say, okay, all the water's gone, let me in. And they let you in on the other side, right? Your cells do kind of the same thing by wrapping their membrane around things and then bringing them in. Because if they just opened up and let a whole bunch of stuff in, what would happen? All its innards would spill out and it wouldn't work very well, correct? So this is vesicular transport. During vesicular transport, we are going to create a package around whatever it is we're bringing in. We can also do this on the opposite end to take things that we've made inside the cell and allow them to leave. Who didn't eat breakfast this morning? Oh, you had a sandwich. There you go, finally. Who didn't eat? No, not counting. So your cells, those pancreas cells we keep talking about, the needs to help you by making glycogen, or glucagon, to break down your glycogen so you don't die. How does that hormone leave the cell? Does the cell open up and spit it out? No. So they create packages or vessels. When I take something into the cell, we call it what? There's a special word. Begins with an E. endocytosis. When I make something inside the cell and I have to spit it out, what do we call that? Exocytosis. Transcytosis. What's that? Oh, if I have to deliver packages to different places in the cell, remember inside my cell, my analogy of the teddy bear factory. If I get a shipment on the dock of eyeballs, they have to go to the eyeball room, right? Somebody's going to take the package to the eyeball room. When we bring something in, we might have to break it down or alter it, and that's transport within the cells. This is going to allow the cells to do all of its wonderful things. Sometimes we can have these packages that form with the help of different proteins. And what we see here is a protein called clathrin. Clathrin-coated vesicles are little proteins that are going to sort of help pull that membrane down to suck things in, help the cell to do what it needs to do for endocytosis to take place. Sometimes when I bring stuff in, I have to break it down. And the other thing we see in this diagram is something called the lysosome. What's lyse mean? Break things down. These chemicals can help break things down. Things like, I don't know, maybe a nasty bacteria. And it doesn't necessarily have to be nasty. It could be chemicals that I make that are really big that need to be broken down into smaller bits before I get rid of them. So lysosomes contain chemicals that will break apart molecules. Do you understand why they're in a package inside the cell? Say I needed hydrochloric acid to break something down in the factory. Should I just kind of spread it all over the hallways and hope that whatever comes in contact with it gets broken down? Would that be very good for the factory's production? No, because it doesn't know the difference between the stuff it has to break down and everything else. If I just let it run around in the hallways, what's it going to do? It's going to break everything down that it comes in contact with. So we have to package it up so it doesn't destroy the cell. So some of the different ways we create these packages and bring things in and have cells then exit with whatever it is processes like phagocytosis. And phagocytosis, with the help of those little receptors and clathrin, are going to allow larger things to come into the cell, like bacteria. Remember we talked last class about a cell called a phagocyte? Is that ringing a bell? Okay. Those cells are going to help protect you from foreign substances. So when you get a bacterial infection, cells like phagocytes can go try to make sure that you don't get harmed by those bacteria. And they'll, through phagocytosis, bring the nasty things in and then do what? Lyse them. So they'll fuse them with nasty chemicals and create a package that's going to break those nasty critters down. Now I have the nasty critters all broken up in a package. What am I going to do with them? Spit them out. Exocytosis. Okay? Pinocytosis. Sometimes I need to get larger amounts of fluid and electrolytes into the cells.
So I just have one question.
Yes, sir.
So when phagocytosis happens, even though that material that is taken in might be bad, it doesn't use it, it just breaks it down and spits it back out?
Yep. For the most part. Because think of it, it's kind of like the waste now, trash. So the systems in my body that take care of waste products for me have to take care of it now. If it's stuck inside the cell, they're not going to be able to do that. What are some of the systems that need to help me get rid of waste products? No. Urinary, respiratory, yeah, circulatory is going to help deliver it. Yeah, integumentary. Digestive system, what's that for? Taking big things, making them small. That's what it's for. The waste that's left in your digestive system is everything you couldn't do what with? Make small enough to use. So that waste is a little bit different. The waste of metabolism, the waste of stuff like this is taken care of by all those other guys. Does that make sense? Pinocytosis is basically the same thing as phagocytosis, except we're talking about fluid coming in. So sometimes phagocytosis is called cell eating, and pinocytosis is called cell drinking. When certain molecules come in contact with very specific receptors on surface of cells, it can force package formation to occur as well. That's called what? The bottom one. Receptor mediated what? Make me a package, bring it in. Endocytosis. So these are different ways of what? Or different forms of what? Which is what kind of transport? Active transport. So these are different forms of active transport. Different ways of bringing things into and allowing things to leave the cell. There's actually a photomicrograph of exocytosis. That's so cool, isn't it? So whatever's in that little package that was created is now being spit out. Do you see why now I don't have a big honking hole in my cell? Because this portion of the vesicle, which is like the plasma membrane, has fused with the actual plasma membrane here. Can you see that? Same thing on the other side. So whatever gets spit out, now this will now become portion of the plasma membrane and not leave a big gaping hole in the side of my cell because I had to deliver something outside the cell. That's extremely important, and that's what the rest of the diagram is going through, the little steps involved in creating the packages, fusing them with the membrane, and then spitting everything out. So think of getting into a submarine and getting out of a submarine. Same thing. If I want to leave the submarine now, I have to go opposite of what I did before. I have to get in the little compartment, lock the door, and then open the other side. What's going to happen? Water's going to rush in, but it's only going to rush into that little compartment and not into the entire submarine. So, endocytosis, exocytosis, pinocytosis. We should understand what those mean as well. Some of the active membrane processes on page 78 in your textbook, table 3.2, are put in smaller terms for you. One of the things you should take advantage of in your textbook is tables, because it takes lots and lots of paragraphs of information and puts them into small. Your goal is to take all of the big stuff and make it small. Yes? Because when you go to study, should you pick this book up and read it? No, because you know what's going to happen? Your brain's going to go, right? Too much stuff. But if you take all the stuff and make it into a smaller form, when you go to study, it's going to make more sense. Yes? So make the big things small. You might have to go check something in the book that you don't quite understand in your small bit of notes. But when it comes time to study for an exam, a couple of pages, 3, there you go. Right? Words like and the should not be in your study notes. They just take up too much space. Right? So words with maybe 2 words beside it. That's what should be in your study notes. So that's what your job is when you study. Bring all that information down to a place where now you can understand it in a small bit. So, we talked about the plasma membrane. We talked about all of the goodies that are in the plasma membrane. The next thing the book talks about is something called membrane potential. Your plasma membrane is polar at rest. What the heck does that mean? Your resting membrane potential is a polar membrane. Now, what's that mean? Yeah, we're talking about the charge associated with the membrane. So again, I emphasize at rest, and that's why sometimes it's referred to as resting membrane potential, because when we talk about nerve impulses, for example, and the nerve is not resting, it is active, we're going to see a different picture. But for the most part, we're going to see a membrane that looks like this with an ion distribution that kind of looks like this. And what's an ion? A charged particle, right? What's an anion? A negatively charged particle. What's a cation? Positively charged particles. So we're going to see that the majority of particles that hang right on the outside of membranes in the fluid that surrounds them tend to be more positive ones than they do negative ones. On the inside, we have some positive ones, but we also have tons and tons of negative large molecules like proteins. So the charge associated with the inside of a membrane, its resting membrane potential, is about what? About minus 90, minus 75 is another number we'll throw around when we talk about neurons. A millivolt is a what? A measure of electrical current. Yes? Yeah, and milli is what? 1/1000th. So when we talk about electricity, like in the wall, it's 1/1000th of a volt. A lot less than what you're getting out of the wall, but still an electrical event. Because of those sodium-potassium pumps, we set up this membrane potential. And we're going to see more positive ions hanging on the outside than we do on the inside. They don't really give us a number because it depends on where we're talking about. So that number varies quite a bit. But it's in the positive millivolt range rather than in the negative millivolt range. Yes? So that is your membrane potential. Again, why do we call it polarized? Yeah, opposite ends, just like the North and South Pole. Just like water was a polar molecule. Why was water a polar molecule?
Because the oxygen was negative.
That end where the oxygen was slightly negative, and the hydrogens that hung on the bottom were slightly what? Slightly positive. This is going to help set up all sorts of interesting things with respect to movement of materials across the plasma membrane. The plasma membrane is selective. What's that mean? Does it let just anyone in? Anything, any size, any charge? No. Based on what we've discussed this whole time, a lot of different things are going to determine whether or not you get in. Do you have a receptor? What's your charge? How big are you? How small are you? Are you hydrophobic? Are you hydrophilic? So there's a lot of different factors that are going to determine whether or not you get in or whether or not you get out. So active transport is going to help maintain all of these electrical charge associated with chemistry gradients that the cell has set up. The next thing the book talks about is cell-environment interactions. Because you are a multicellular organism, you are made of more than one cell. Your cells need to have a highly organized way of communicating with each other, because tissues and organs are what is going to help you function as a human being. So many cells in groups have to work together to help do what they need to do for you as a very highly complicated multicellular organism to maintain homeostasis in your environment. Yes? So we threw a lot of words that you know, right? At you, hopefully. Hopefully you know those. First one they talk about is CAMs. Cell adhesion molecules. We talked a little bit about this earlier when we talked about the connections between cells. Very important to hold cells together. But not only to hold cells together, but to also signal other cells as to who I am and what I do for a living. Some other receptors are talked about under the roles of plasma membrane receptors on page 81 in your text. So we talked a little bit about this in last class as well. Some of them act as signals to other cells, recognition for other cells, like little signs stuck in our plasma membrane saying, yeah, I belong here, that cell who's responsible for my immune system, so go on by. Receptors for certain chemicals, like hormones. And what we see here, let's pretend that little ball in the diagram is a hormone. When it comes in contact with these receptors, it can cause the cell to do something different. It can cause the cell to change what it does. It's sort of like the call in the teddy bear factory from the boss that says, hey, shipment is due. You guys are a little slow. Get a step on it. Does the boss ever come into the factory? No, some bosses do, but in this case, no. He's going to call over the loudspeaker. Hurry up! Move it! And what's going to happen inside the cell? They're going to pick up the pace. So a lot of these receptors and chemical messages can change what's going on inside the cell. We're going to hear this a lot again when we talk about the endocrine system, second semester, when we talk about hormones and how they affect cellular activity. This is an example of something called a second messenger system, where I have some sort of chemical on the outside of a cell, and again, that's our little ligand, that comes in contact with a receptor on the surface of the cell, that then sets up a series of internal changes within the cell. In this case, when chemistry happens, molecules change shape. And the change in shape might kick something else off in a chemical reaction. Sort of like when you line up dominoes, right? What's going to knock all the dominoes down? The first one changing shape, smacking into the second one. Then the second one smacking into the third one, and so forth. So many of these things are sort of like chain reactions that go on within the cell. And they get set off when a hormone or a ligand comes in contact with a receptor. Remember, that lock and key thing is going to come into play. It's going to be a very specific shape, and the receptor has to have that shape to accept it in order for it to get set off. Inside the cell, we're going to see something called the G protein in a second messenger system. That's then going to cause a series of changes within the cell that will cause the cell to do something different. In this case, in this little cascade, it talks about activating kinase enzymes. And in this case, those enzymes, now more abundant in the cell because of this little chain reaction, are going to speed up the effects of those enzymes, whatever those enzymes' job was. Just like in the teddy bear factory. Speed it up. We got a shipment. So cellular mechanisms will speed up. What caused that to happen? Yeah, the ligand, the chemical, coming in contact with who? The receptor. Okay? You with me? I think I'm losing some of you. Some of those channels, I should go back to the picture. Some of those channels will cause changes when there's a change in voltage, when there's a change in charge. Remember we talked about that sodium-potassium pump? That job it is to move things against the concentration gradient? Well, when that guy gets paralyzed, and we'll talk about that during a nerve impulse or an action potential, sodium is going to rush in and cause a change in charge within the cell. That change in charge can also cause these proteins to kick in chemical reactions as well. So that's what they talk about under the role of voltage-gated membrane channels. Again, we're going to talk about this in much more detail when we talk about what? Nerve impulses. All right, the cell. What's a cytoplasm? Fluid where? Inside the cell. Is it like water? What's plasm? It's kind of like water, but it's thicker than water. It's kind of like runny jello. Still a lot of water there, but there's a lot of other things like proteins as well. The other thing we're going to find inside the cytoplasm, beside the liquid stuff, cytosol, is a whole bunch of little parts. Guess what we call the little parts? Organelles. That means little organs. By the way, before I forget, did somebody leave their clicker in this classroom last time we were here? No? Okay. It might have been him. I'll leave it on the bench. I'll leave it there. So organelles, little parts, little divisions inside the factory, little divisions that do different jobs for the what? For the whole cell. We also see what we call inclusions, different chemicals, different proteins that are going to help the cell function. So the book then goes on to describe all of the little parts of a cell. And basically what you have to know from this chapter is a definition of what all the little parts do. You have to visually be able to identify them, and where you're going to be doing that? In lab. So remember that generalized cell I showed you? Remember I said you're going to see that diagram again? Where are you going to see it? Lab. What do the parts do? This part here is called the mitochondria. Sometimes it's referred to as the powerhouse of the cell. How come? What's it doing for me? This is where The majority of ATP is produced in a chemical reaction called cellular respiration, which we will cover next semester. This is where the big kahuna of ATP is made, during that reaction. What's a ribosome? Ribosome is two little components of nucleic acids. What's a nucleic acid? Yes, phosphate, sugar, nitrogenous base. What's that? Those little units. Oh my God, I have to remember this. Nucleotides! Ribosomes are a form of nucleic acid, ribonucleic acid, that are going to help me put together proteins inside the cell. Sometimes we'll see it referred to as the site of protein synthesis. And we're going to find them all over the place. But a lot of them can be found on these little canals that we see here. The next organelle they describe in the book is called the endoplasmic reticulum. And what's that doing for me? Yep, transport. Now we'll notice that that big honking purple thing with the yellow things on it, what is that? That's a nucleus. And our nucleus, because we have what kind of cells? You have what kind of cells? Eukaryotic cells. Our nucleus is wrapped up in a plasma membrane of its own called the nuclear envelope. But there's big honking holes in it, yes? Because information needs to come in and out. And guess where the information coming out of the nucleus is going to travel? The endoplasmic reticulum. And some of that information is going to help us build proteins. Guess what the rough endoplasmic reticulum has all over its surface? Ribosomes. We are also going to find areas that don't have as many ribosomes on them, but are still important for transport, called what? Smooth endoplasmic reticulum. So we're going to find altering of chemicals and proteins happening inside the endoplasmic reticulum system. All right, say I made that wonderful hormone during protein synthesis in my endoplasmic reticulum, and I have altered it through the smooth endoplasmic reticulum, and now it's time for me to have it packaged up to leave the cell. Who's going to help me package and ship inside the cell? The Golgi apparatus. Remember, I have to create a membrane around something before it leaves the cell. I call the Golgi apparatus the UPS system of the cell. They package and they ship. So the Golgi apparatus can package things up, and through what process have them leave the cell? Exocytosis. It can also make little vessels to hold things like those lysine chemicals. Another group of chemicals that can help me break things down in the cell that I'll make and package up is peroxisomes. Peroxides, excuse me. Peroxisomes are the packages that Golgi makes for me to hold those peroxides. Do you ever see what happens when you take hydrogen peroxide and pour it on a cut? Have you ever done that? Because you cut yourself and your mother goes, "Come in the house, we need to disinfect you." Right? What happens when you pour the hydrogen peroxide on the nasty cut? Bubbles. The bubbles do what? Pop. Imagine if you were a tiny little prokaryotic bacteria on the surface of the boo-boo. And the hydrogen peroxide hits that cut and creates bubbles. It's kind of like walking through a minefield for bacteria. When the explosion happens, it's going to do what? Explode them. So peroxide-like materials are housed in little packages called peroxisomes. We're going to find a whole bunch of different packages inside the cell. That general term for a package inside the cell is a vacuole, and it can house all kinds of stuff. It can be a water vacuole. If I look at plant cells, for example, I find very large ones that actually give the plant its shape. I can find peroxides in it, in a peroxisome. In a lysosome, which is the same thing, another little package, I can find different types of chemicals that are going to help break down different things within the cell. So the cell has what we call an endomembrane system. And all of these things I'm talking about are bound by what? Who? Who? Plasma membranes.
Did you want to click? Or we have to go?
Oh, you gotta click. Yeah, everybody hold on. Click for attendance. Adios. Is everybody clicked? Nope, still got people clicking. So keep clicking, I'll keep talking. Along with the endomembrane system, membrane-bound organelles, we're going to find tons and tons of different protein structures that are going to help give the cell shape. They're going to help with transport inside the cell, and that's called the cytoskeleton. Is everybody clicked? Yes? Say yes. What did everybody pick? I always love to see what everybody picked. Oh. Everybody likes A. Let's say A is the right. No, let's say D is the right answer. Okay. So the cytoskeleton, that's not a right or wrong. That's just me digressing. Is also very important to understand. There are different thicknesses of protein structures that are going to help with the shape and transport inside cells. Microfilaments, little ones. Intermediate filaments, the in-between guys. And the other guys that are big, thick, kind of like telephone poles, are called microtubules. By the way, this is a cute picture of the chemicals lysosome inside cells. So we have to understand the definition of these wonderful little protein structures that are going to help with different functions inside the cell. We're going to talk about each of them when we talk about different functions within the cell. But they all kind of have the same structure. Some are more complex than others. Some will help different things happen within the cell. Things can move down them and move across them during cellular reactions. And they also can band together in these huge microtubules and actually help the cell to reproduce itself. Many of these microtubule arrangements can be seen, depending on what I do for a living, in many different places in the cell. When I have to split, we're going to see it inside those centrioles we just passed by. When I have to move things across my surface, we'll see them organized in these little finger-like structures called cilia. that sort of wave back and forth in fluid to help move things across the cell. We'll see that same sort of structure in cells that have to swim from one place to another, that have something called flagella. And these little wave-like motions can help keep homeostasis by moving things across the surface. So cilia and flagella are a part of the cytoskeleton. They're little extensions sometimes of the plasma membranes with these very complex proteins in them. Extensions of plasma membranes can also be described as something called microvilli. Little what? Little fingers that can help move things across cells. Or increase surface area. The heck is that supposed to mean?
Like the main code.
Yeah. So if I take my microvilli, pretend my hand is a cell. And my job is to absorb things on my surface. Do you see how I've now created a much larger surface area in a smaller space? If I took my fingers and I drew a line with string across them, and then I took the string off and pulled it out and straightened it out, it would be this big, right? But now I can do all of that same work with my little microvilli in this tiny little space. Okay? So that allows for greater surface area, movement of things across the surface to help with cellular function. The largest of the organelles is the nucleus. That's pretty much the only organelle you can see clearly with your compound light microscopes. All the other guys are going to look like little dots like sand. But you can see a nucleus, right? Those of you who had lab already this week, you can actually see the nucleus dividing. It's so gosh darn big. But when you look at a cell, most all cells, you see whatever shape the cell is and a big honking dot in there somewhere. The big honking dot is the nucleus. The nucleus is what contains what? Your map. What is it? DNA. Deoxyribonucleic acid, right? Some cells are multinucleated. They're so giant they need information down the line. When we talk about skeletal muscle cells and we look at it under the microscope, it actually looks like striped telephone poles. So those huge cells have more than one nucleus. But most cells have one. Some have a couple. We even have anucleated cells in our body. We talked about those a little bit already. Who are those guys? Yoo-hoo! Remember red blood cells? They don't have a nucleus. When they're born, they have a nucleus. They're born, anybody know where red blood cells are born? Red bone marrow. But by the time they squeeze their way out into the circulatory system, they spit their nucleus out. So can they divide anymore? Nope. So those are anucleated cells. We find those in human as well. The plasma membrane associated with the nucleus is called the nuclear envelope. It's a double membrane. And it's got big pores in it called nuclear pores. Nuclear pores are important because we need to transport some of the information we get from genetic material outside the nucleus to the cell so they can do their job. Think of the DNA inside the nucleus as the big boss. And we have molecules that need to get information from the big boss and bring it out to where all the work gets done in the factory. Another thing we're going to find inside the nucleus is something called the nucleoli. And there we're going to assemble some very important nucleic acids for use within the cell. Ribosomal RNA, for example, is going to be assembled there in the nucleoli. And then where is it going to go to build ribosomes? Where are ribosomes located? Outside the nucleus. How is that information then going to leave the nucleus? Through the nuclear pores. Yes? Travel down the endomembrane system, the endoplasmic reticulum, and assemble themselves on the surface of a rough endoplasmic reticulum. Yes? You with me? So this is a picture of something called chromatin. In order to get the information I need from the map, I have to be able to open up the map and read it. The map is in the form of a double-stranded nucleic acid called DNA, assembled in a very specific order. And what is the specific order I'm talking about? Specific order of what? Adenine, thymine, guanine, cytosine. What were those guys called again? Nitrogenous bases. Very good. Nitrogenous bases. And the order of those nitrogenous bases is your map. You've got tons and tons and tons and tons of nitrogenous bases that make up your genetic material. Now, in order for me to make a new cell, I have to have all the parts, and I have to have a map. So during a cell's growth and reproductive life, tons of different things are going on there. In some cells, not all cells, in the beginning all cells, how many cells did you come from? One. You started as one cell. Like, wait a minute, there's two cells. Yeah, okay, if you want to get technical. There's two half cells that come together to make what? Your one. But you begin as one cell. How many cells do you have now? More than one. Right? So that cell that you started out at was a cell that had the map and all the parts. Now I got to make a whole hell of a lot more of them to make you, right? And through that process called mitosis, we are going to make a whole hell of a lot more cells in a multicellular organism. So the life of a cell, once we get past that stage of crazy making more cells during your incubation period, this process is still going to take place later on to regenerate old tissue, replace old tissue once you get to the adult form that we see now. So when we talk about the life of a cell, it involves different stages. This is called the cell cycle. Now, if I'm just hanging out, and sort of think of your life as kind of like this. When you were born, what did you do after you were born? And luckily for your mother, you weren't this big when you were born. Yes? So, again, I will ask the question, when you were born, what did you do after that? Yeah, you had to get bigger. You had to grow. Think of that as the cell's interface. So when a cell is first born, it has to go through a period where it gets to the size it should be and gets all of the organelles that it should have in order to function properly. Kind of like when I first start up the teddy bear factory. I got to make sure I have all the parts, I have to hire personnel, I got to do all that stuff. And then I do what I need to do for a living as a cell. That's what goes on in the G1 phase of the cell cycle. At some point, I decide, well, you know what? It's probably time to make more of me. Because I'm starting to get a little old, and I probably could use a refresher. That's the S phase. In order to make another one of me as a cell, I have to make sure I pass on the map. The entire map. Every single page of the map. Where's my map? Inside that DNA. So during the S phase of my interphase, my doing what I do for a living as a cell, when I decide it's time to split and make two new, I have to make a complete copy of all the genetic material. So I have to then open up all my genetic material, make a copy, and now that genetic material, which was located in what we call chromatin, is copied and stuck together in what we now call chromosomes. When you move from one place to another, what do you do with all your stuff? You just kind of throw it all in the car, head down the road. What do? You pack it up. Why do you do that? Make it easier to move, and that way you won't lose your stuff. Yes? Well, sometimes that doesn't happen either, but that's beside the point. That's what the cell has to do once it makes a copy of the 42 chromosomes. How many chromosomes do you have? It's not 42. 46. 46. You're just throwing out numbers. 46 chromosomes, right? Once I'm at the end of replicating, how many do I have? Do the quick math. 92. Holy crap, it's kind of crowded in that nucleus now. It was crowded before, and now it's really crowded. So I definitely have to package it all up and get ready for the move. I'm going to wind it around little proteins called histone proteins, and that's what's going to go on during the S phase of interphase. For a brief period, I'm going to make sure everything's going well. Make sure my apartment's ready on the other end, make sure everything's in a package, and that's what's going to happen during the G2 phase of interphase. So you prepare for division. Make sure everything's packed, make sure everything's good to go. And once you've gotten past that stage, you're ready for the next move in your life. You're ready to make a new cell. The next point is what we call the mitotic phase. So most of the cell's life, for the most part, and again, it depends on how fast I'm dividing, is spent in interphase. Mitosis happens pretty quick. And the mitotic stages, you should understand what is going on there. Now, with the help of a whole bunch of enzymes, I can replicate my genetic material. Enzymes that are going to help me make more nucleic acid are called polymerase enzymes. And when I'm making more DNA, guess what we call the enzyme? That helps me make more DNA. We call it DNA polymerase. So we're going to use the original DNA to create a copy on either end. We're going to unzip it in the middle, and with the help of DNA polymerase enzymes and all of these free nucleotides floating around that you have from, where'd you get free nucleotides floating around? How do you have all these nucleotides? Where'd you get that building material from? That was a hint.
Water.
No. There's more than water in there. There's some sugar and there's some cream. You get it from other nucleic acids that you ate. When I put them in here, they're big, yes? And then what am I going to do? Break them down in the digestive system. Then I'm going to take the parts and that's where they came from. So I have a whole bunch of free ones floating around, and I'm going to use those to help me build a whole nother copy of genetic material. With the help of DNA polymerase enzymes, I'm going to use those free nucleotides to replicate my genetic material. And I'm going to make a total copy during the mitotic stages. Well, I'm sorry, I do that during interphase. So interphase isn't just me sitting around doing nothing. And cell division is mitosis. This is where I'm going to take all that stuff that I created, and I'm going to start to separate everything and split. Now you guys who have already had lab, you went and actually looked at this, because you can actually see this under the microscope. Because after I take my 46 chromatids and make 92 chromosomes, you can actually see them. They're so thick. Because when I package them up and twirl them around those histone proteins, they actually look like little what? Little black finger-like things inside my nucleus. So when I start packaging everybody up for the move, that's the first stage of mitosis. That stage is called what? No. Interphase happens before. We have to prep before we move, right? Mitosis is the move. Prophase. Prophase is when I pack up all that genetic material and you start to see those little black finger-like structures. The other thing, in order for me to pull my copies apart and make two brand new cells, that has to happen at the end of prophase, what do I have to get rid of? That nuclear envelope. It's just going to get in the way. So I'm going to break it down. I'll use it again. I'm pretty efficient. But I'm going to break it apart. So by the end of late prophase, we've gotten rid of that nuclear membrane. We've packed up all the goodies. They've already been copied in the S phase of interphase. And now I'm ready to organize everybody for the move. Everybody see Toy Story? I love that movie. Get a moving buddy, right? Make sure you have your pair. Well, there's certain proteins that are going to hold together those copies you made of your genetic material. And in order to make sure that everybody gets, that means the two new cells that are made, gets every single portion of the map, the best way to do that is to line all of those chromosomes up in the middle of the cell. That stage is called what? Metaphase. Now how am I going to pull apart those awesome pairs of all that genetic material? The centrioles are going to help me out with some microtubules. They're going to create these little sticky spindles. If you look at the picture, it's the green stuff in the picture. And that's kind of like a fishing rod. Whoosh. When you fish, does anybody fish? How do you catch a fish?
Patience.
Patience, but you got to have an instrument. What is the instrument? The fishing rod. Fishing rod, and what comes out the end of the fishing rod? Some sort of wire, something or other, and you put a little hook on the end, and you put some delicious food on the end to catch a fish, right? Think of those spindles as a fishing rod. And the hook is attached to what? one of the pairs in that chromosome. During the next stage of mitosis, I'm going to reel in the fish. So those wires, those microtubules, are going to start to shorten up back into the centrioles where they came from and pull apart what? Pull apart those pairs of chromosomes that I made. Pull them to opposite sides of the cell so that when it comes time to make two new ones, each cell, two new daughter cells, will have a copy of that genetic material. During the end of the next stage, and again, that pulling apart stage, that's called anaphase, pulling away from each other. What else does a cell have besides chromosomes? Do you know? What else does a cell need to be a complete cell besides the chromosomes? That's important for me. Wait a minute, I got something interesting for you. It's going to take me a while to get there, so I'll keep talking. It needs all the other parts, right? The process of replicating all the other parts, which starts at the end of anaphase and continues to the next phase of mitosis, which is called what? It's called telophase, is a process called cytokinesis. What's cytokinesis? Nope. Cytokinesis is, damn it. It's the replication of what? The cell's organelles. So that's going to start at the end of anaphase, and it's going to continue on through the end of telophase. So when you see telophase described, it's telophase and what? Cytokinesis. Know what cytokinesis means? So that's the nucleus. Remember, I got to make all my other parts before I split. Cool, huh? Listen, I didn't finish, but guess what? You're still having a quiz. All right? So you're going to have a quiz on Chapter 3 on Tuesday. We'll finish the very end. The very end is the whole protein synthesis thing. And what is going to happen on Thursday of next week? You have your first exam, chapters 1 through 3. So you have a busy weekend in store for you. Adios. Oh, geez, hold on one second. Yeah, I fixed it. So we're good. I fixed the attendance too. Yeah, you'll get my message. Clicker, clicker. Hi, dear.